Hello and welcome to today's webinar entitled Purpose-Grown Trees for a Sustainable Future. I'm Dan McClellan, e-learning coordinator for the BioNetwork BioEd Center, and our BioForum webinar is scheduled to run from 11 a.m. until approximately noon Eastern Time. Our panelist today is Maud Hinchee. Maud is the Chief Technology Officer for ArborGen, and in just a moment I'll turn it over to Maud and we'll begin today's BioForum. But first, we'd like you to feel free to ask questions by typing them in the chat box located on the right side of the screen. I'll be keeping a record of each question that is submitted, and we'll have a time at the end of the presentation to address all of your questions. And be sure to send your chats to all participants, and this will ensure that we don't miss your question. We'd like to know who we have in our audience today. I've loaded a poll, and you should see it on the right side of your screen. So please select the option that best describes you. Also, we want to have an accurate record of your participation in today's BioForum. So for question number 2, let us know if you are sharing a screen by typing the total number of participants at your location. Now, if you're the only person viewing at your screen, then just type the number 1. And while you're doing that, I encourage you to mark your calendars for next month's BioForum. Information and registration details are available online at ncbionetwork.org. At this time, we'll begin today's BioForum, and I'll turn it over to Maud.
Thank you very much. It's a real pleasure to be able to present to you a little bit of the work that we're doing here at ArborGen in the area of advanced technology for forestry. So the title of the presentation today is Purpose-Grown Trees for a Sustainable Future. Before we begin to talk about the technology that we're going to be discussing today, I just wanted to mention that today's forests are in a point of crisis, and the World Wildlife Foundation recently released a report called the Living Forest Report. And in it, they indicated that basically by the year 2100, the projected forest area was going to decrease dramatically, and that it was changing very quickly. And so that it's very important that we understand what it really will take in order to achieve secure and healthy forests. Because in the past, secure and healthy forests have helped stabilize the climate throughout the world. They're very important for that. And it's been found and reported in the WWF Living Forest reports that responsibly managed plantations actually can perhaps in the 7% on which they're planted, be able to supply 2/3 of the forestry needs that will be needed by the 2100 time period. So it's important that responsibly managed plantations continue to supply fiber and for materials and energy, and to deliver important ecosystem services, and then also be able to provide the landscape that provides the economic basis for towns, productive farms, and nature reserves, as well as being able to help maintain wild forests. So maintaining forests is a cornerstone of national and international policies, and actually part of ArborGen's vision statement. In the Living Forest vision, it's really about how do you halt forest loss, but balance that with the potential implications for human well-being, economic development, and the wider environment. Currently, the people on Earth are exceeding the Earth's biocapacity to produce renewable resources and absorb CO2 by 50% or greater. And basically, in order to kind of stave off this ecological overshoot, we need to balance human demand with the regenerative capacity of the planet. If we don't step in and create more productive forests that are sustainably managed, then with the current resource use, we will need the equivalent of 2 planets by 2030. So the goal that WWF put forward in their Living Forest Report is to sustain a net zero deforestation by the year 2020. That's the goal that they've set. And what they've said in that model is that achieving net zero deforestation by 2030 will require higher productivity across large, often suboptimal areas of land. It will require hundreds of millions of farmers and foresters to change to more sustainable and productive practices. And that, in the time frame that's going to be required, that's a task that has previously been unprecedented. Improved productivity is important, but we have to be aware, and as ArborGen is, that improved productivity can bring its own environmental costs. So we have to be aware of the impact of more highly productive forests and actually mitigate that in our management practices. And those possible environmental costs could be salinization, erosion, depleted aquifers, increased energy use, and pollution and biodiversity loss. So it's really important that technology advance in the areas of low input, knowledge-based intensification that utilizes the best technology to be able to get the highest amount of productivity per acre of land with the least negative environmental impacts, and that meets our sustainability objectives. And it's our belief at ArborGen that tree genetic improvement, whether it be through traditional breeding or through biotechnology, actually has a role to play in achieving net deforestation. We believe to meet the demands on our forest resources, we will need to maximize the efficiency of the existing resource areas for managed forests, managed plantations. And as stated in the FAO document on biotechnology, that biotechnology provides powerful tools for the sustainable development of agriculture, fisheries, and forestry, as well as the food industry. When appropriately integrated with other technologies for the production of food, agricultural products, and services, biotechnology can be of significant assistance in meeting the needs of an expanding and increasingly urbanized population in the next millennium. And in fact, as reported by ISA in their global status of commercialized biotech and GM crops in 2010, since biotech plants were first launched commercially in 1996, there's been a significant adoption of these crops that have been modified through biotechnology, where now there's over 29 countries in the world that grow these crops, over 300 million acres planted in 2008, and continuing on. I think it's 3.4 million now, and greater than 2 billion cumulative acres planted, and the percentage of crops that are now biotech throughout the world is 81% of the soybean crop throughout the world is biotech, 64% of the cotton crop, 29% of the maize crop, and 23% of the canola crop. And in these different world areas, the regulatory pathways are in place in these key markets and in the developing world, and there is growing, increasing public acceptance of biotech crops, such as what we see in Brazil and Latin America. Tree biotechnology itself is the application of biotechnology to commercial plantation forestry, and it offers many opportunities to increase the productivity and the quality of the crop. Biotechnology contributes to the goals of producing more wood per acre. It can be used to improve forest health. It can be used to aid sustainability practices and to lower greenhouse gas emissions. If you can achieve increased productivity per acre, forest biotechnology will play a significant role in meeting these global mandates. And basically these drivers that we see now that are causing for a greater amount of forest, greater amount of productive forests are the drivers of renewable energy, climate change, and achieving food security. ArborGen is a company that's focused on the future of forestry. We're currently a leading producer of purpose-grown trees. We sell more than 300 million seedlings a year. Our base business is the feed planting stock right now that is planted widely for reforestation as well as for the pulp and paper and lumber industries. Within the company, we have over 50 years, much over 50 years of experience in forestry and technology that's related to forest improvement. We're comprised of a multinational team of dedicated conservationists, biologists, foresters, researchers, and scientists. And we are a technology leader in that we have innovative product platforms in which we're using advanced traditional breeding methods as well as biotechnology to improve pine and hardwoods. And we have, through our genetics improvement program and the diversity of the genetic resources that we have within our company, we are able to create a pipeline of world-class high-performance germplasm. And we are the most active forestry company in the world in terms of field trials and research, and we have more forestry field trials than any other forestry company in the world. And one of the things ArborGen is focusing on is addressing the demand for woody biomass, because we see the demands are increasing as we see these new markets are emerging. The pulp and paper and wood products industry is the traditional industry that the forest industry has been focused on in the past, the forest plantation industry. But now there are new activities in the area of generating electricity, whether through direct firing or co-firing in coal plants, or in developing advanced biofuels where woody feedstock is used in the conversion into bioproducts that can easily be converted as intermediates towards biofuels or other advanced bioproducts. So basically the demand for renewable feedstock to replace petroleum-based or non-renewable products is growing. And we need to address that through genetic and technology improvements in order to meet the demand. And ArborGen has multiple different ways of approaching that, from basic breeding all the way up through the biotechnology. So we believe that the world doesn't just need more trees, but we need trees that can do more. So our approach is to provide better, more sustainable, purpose-grown trees. We have a strong conventional tree improvement program in which we do breeding and selection and use advanced technologies such as the genomic sequence and molecular markers in order to assist us to be able to do the best combining of parents to produce the best progeny. And we also have accelerated improvements through advanced genetic technologies through the use of hybrid technologies where we're crossing different species in order to get a better performing tree, and advanced propagation technologies in which we can identify superior genotypes that can then be propagated at a larger scale, and then introducing traits through biotechnology. So basically ArborGen's current products are basically we supply for planting and reforestation and the traditional pulp and paper industry more than 100 species of pine and hardwood, which includes loblolly pine, slash pine, logleaf pine, oak, elm, birch, poplar, and black walnut. Breeding supports and biotechnology work supports an advanced product portfolio that's directed towards eucalyptus, loblolly pine, and populus. And in our research program, we focus on three basic categories. We have the category of traditional breeding and genetic improvement, which is called improved germplasm. We have research that looks at the silvicultural practices in order to optimize the productivity with the least input cost and the least unnecessary use of chemicals through the management systems. And then we have the biotech improvements. And in the improved germplasm, we're focusing on conventional breeding and varietal technology in which we identify superior genotypes that can provide significant value. For example, a pine tree that has the perfect form to be the best, most superior tree for lumber, you can identify those particular trees and grow them and then increase the value for the landowner in terms of the amount of lumber producing trees he's able to generate on that acre versus pulpwood trees. In the management systems, we're looking at increasing densities, shortening rotations, and doing other silvicultural improvements relative to fertilization and weed control. And in biotech improvements, we're focused on improved growth, shorter rotation, stress tolerance, improved processing, both from a biofuels perspective as well as from a pulping process, and improved wood quality that would be directed towards improved structural lumber. So productivity improvements that we're talking about here actually would allow the processor to require less land that's producing the wood that they need to feed their plant. In other words, productivity improvements decrease the amount of land required to supply a ton of wood for the processor, either in the traditional forestry or in the future bioenergy industries. I'd like to go in now and talk about a specific example of how biotech can provide a solution to meet an unmet need. In the production of renewable products such as lumber, pulp, paper, and bioenergy, it's been discovered by those that are in the processing industry that at times hardwood in the southeastern U.S., hardwood being, things such as oaks or sweet gum, and maple, those might be limiting. In other words, sometimes they're not available in close enough proximity to a processing center. So what happens is the price and difficulty of getting that wood affects those processes, so they can't be profitable, and sometimes they even have to close down the mills. What we're seeing in the traditional pulp and paper industry in the southeastern U.S. is that at times they have to actually buy pulp from Brazil or from other countries, or not pulp, but wood chips, because they don't have the hardwood available at a price that's acceptable in the U.S. If you can take a look at the graph that's on the left here, you can see that this graph right here is called the U.S. South Private Operable Growing Stock Inventory. That's rather elaborate a way of saying is what's able to be harvested at any given time. And you can see that we're seeing an increase starting in about 2008, 2009 of the standing inventory of pine. However, in the hardwood case, what we're seeing is there's been a decrease in the hardwood inventory, the available wood that's available, the hardwood that's available to harvest at an economically feasible price. And if you take a look at the prices and how it's changed over the time, you can see that the prices of that hardwood are going up. Now, what that does is eventually drive the processors, such as a pulp and paper company, to close down mills because if they're not able to get the feedstock they need to generate the paper that they need at a cost-effective price, then their only option is to shut down that mill. And we've seen that happen a lot in the southeastern U.S., not just for the reason of hardwood supplies, but for other reasons, but it is a contributing factor to that. So what we would like to do is to understand how eucalyptus, which is grown widely in Brazil as a pulp and paper species, how that eucalyptus can now be grown in the southeastern U.S. to meet this demand. And you can see here the Eucalyptus grandis by Eucalyptus Europhila hybrid. They commonly use hybrid eucalyptus in Brazil. It can be grown in the southeastern U.S. in Florida in non-freeze challenged conditions. And you can see after just 4 years worth of growth, there's significantly tall trees that are able to produce 27 to 33 dry tons of biomass per acre per year. And we can see the pulpwood potential is 7.5 to 12.5 dry tons per acre per year. And the trees can, in that 4-year period of time, can achieve being 56 feet tall and about 6.4 inches in diameter. What we're looking at is looking at different species of eucalyptus that are chilling tolerant, and also looking at a biotech application of taking the eucalyptus we saw in that previous picture that grows well in Florida, and being able to add a slight improvement in freeze tolerance to enable it to grow in the coastal regions of the southeastern U.S., which are areas in which hardwood can be limited. Well, we see that these trees have the potential to provide significant biomass productivity. And because eucalyptus can be coppiced, which means you can cut it and regrow it, cut it and regrow it, then you only need to sometimes plant on basically after 3 harvests, probably can only go to 3 or 4 cuttings. So basically over a period of 12 years, you can get probably 2 to 3 crops. So we're looking at a species, chilling-tolerant ebenthami, growing, and this is an example of that tree. It's naturally chill-tolerant, growing in South Carolina. And it has a productivity rate of 14 to 18 green tons per acre per year. We anticipate it'll be able to be grown on a 4-year rotation, and as I mentioned before, have 3 harvests over that period, 3 harvests with the coppicing capability over that period of time of about 12 to 14 years. And in the case of the biotech eucalyptus that we see here and pictured in Alabama, we're anticipating that would be able, because it's growing in a faster growing species, it would be able to produce 14 to 33 green tons per acre per year over a 4-year rotation and 3 harvests. In particular, we're looking at the tests that we've been doing with a particular genotype of the freeze-tolerant eucalyptus, the biotech eucalyptus, which we've given the name of AGEH427. And that line of eucalyptus is based on that hybrid that I showed you on the previous picture, that Urograndis hybrid. It's the same base genotype as that one we saw growing in southern Florida. And it's been genetically enhanced to tolerate freeze events that we have in the south, these kind of erratic winter freeze events that we have. And it can grow. It's currently being grown on a research scale on various sites along the Gulf Coast. And it yields 15 green tons per acre per year within the identified deployment zone, which you can see on the picture here on the right. Basically, the identified deployment zone is this purple region here. You can grow the freeze-tolerant eucalyptus here as well as the chilling-tolerant eucalyptus. In fact, the chilling-tolerant eucalyptus can probably grow a little bit more north, whereas that Urograndis hybrid that is the base clone for the freeze-tolerant eucalyptus primarily grows in central to South Florida. Above this area in the more coastal regions of the southeast, we anticipate that populus will be the preferred hardwood of choice. So with we're anticipating that the AGEH427 will achieve that yield of 15 green tons per acre per year within the identified deployment zone, and that the advantage that eucalyptus has, which the Brazilians have been able to capitalize on, is that it has excellent qualities for pulping and bioenergy, and it's quite suitable for mulch and other products. So it has multiple end uses, and it's an excellent tree for that reason. The freeze tolerance that we've introduced into that AGEH427 is a gene called the CBF gene, and it's a gene that acts like a switch. A transcription factor is essentially a switch that can upregulate multiple pathways at the same time. Imagine a single switch that turns on lights in multiple rooms in a house. And in this case, consider each one of those rooms being the way the plant copes with freezing events. So, for example, plants can respond to freeze by stopping growing. Plants can respond to freeze or tolerate freeze by increasing the amount of solutes in the water within their cells, so that just like if you put salt in water, it doesn't freeze as easily. It can also protect membranes, make them stronger so they don't rupture as much if there's any ice crystallization. So what the CBF gene does is when it expresses in plants, it upregulates or turns the switch on to those mechanisms in the cold protection pathway. It's a gene that's found in all plant species, but in tropical plants, or plants that have been adapted to tropical climates, even though it's there, it has a kind of a dysfunction where there's the circuitry, for example, from the CBF2 gene to these downstream pathways is not working properly. So what we've done is we've used a CBF gene that came from a Arabidopsis, which is a common plant used in plant genomics research, and we've been able to add that back in and shown, as they have shown previously in other plants, that that gene can induce and promote the cold and freeze tolerance pathways in plants that have had that gene introduced. We also, besides using that gene, we put it under the regulation of a regulatory sequence, which is called a promoter, that responds to cold. So in other words, the CBF2 gene now is itself responding to a switch. So when cold is sensed, the regulatory sequence that drives the expression of the CBF2 gene actually turns on the gene, but only when it's cold is sensed. And that's a promoter that's called RD29A, and that also comes from Arabidopsis. So the genes that we've introduced are plant genes, essentially, that we've added to eucalyptus. We've essentially taken a gene that's found in a temperate plant and put it into a tropical plant to generate the response. And this next slide kind of shows the cartoon of how basically the eucalyptus tree will sense the cold, the CBF2 gene will be triggered, that protein itself then binds to pathways or genes that are in the downstream pathways, and they produce products that then protect the plant. So basically, by switching on this one gene, we're able to upregulate multiple cold tolerance pathways in the plant. The way that the gene was introduced into eucalyptus is that eucalyptus is grown and propagated as tissue culture stalks. So imagine you make cuttings of things like philodendrons or camellias or whatever. Basically, you can take those and keep those cuttings in tissue culture, and we use those stalks to use a method that's a biological method called agrobacterium transformation to introduce the genes. And then each cell within those tissues that have been, into which the genes have been transformed, each one of those cells is capable of forming little plantlets, which we then can identify through the tissue culture process, propagate it, and then root them, and then go ahead and test them in the field. So after we introduced the freeze tolerance gene, we first, when we generated the plants, we first did a cold chamber test, where we took trees that were growing in the greenhouse and basically plunged them into a cold chamber, and then brought them out to see how well they did in terms of recovering afterwards. On the left, you can see after that extreme test in the cold chamber that the growing point of this tree that doesn't have the CBF2 gene, that growing point is completely killed, whereas you can see in the one that has the CBF2 freeze tolerance gene, the growing point is continuing to grow and looks quite healthy. So what we've been able to see, looking at the different temperature extremes, is that the control, basically, this is the vertical, the y-axis here is percent dieback. So you have increased amount of dieback at increasing temperatures, and you can see that there's a 3-degree shift here where the line, the 427 line and another line that we looked at, the 435 line, show improved freeze tolerance. In other words, less dieback at increasingly lower temperatures. We have then demonstrated that this freeze-tolerant eucalyptus in the field, we've looked at it at many, many different field sites in many different areas. And you can see in one of the first field trials that we did at this, the control tree, this is just in a South Carolina field trial, you can see the control tree was hit pretty hard by one of our episodic freezes, but the lead line looks strong and green. And then here on the right, you can see an aerial view of a field trial that we conducted in Alabama. And you can see that these are blocks of the same, each block is a different genotype. And you can see these are the controls that do not have the freeze tolerance gene, and you can see the performance of trees that do have the freeze tolerance gene. So we were able to see in this Alabama field trial tolerance to approximately 16 degrees Fahrenheit, about -8 to -9 degrees centigrade. And the height at 4 years is currently 56 feet with a DBH at 48 months of 6.4 inches. We've continued to look at these trees in the field, and we continue to see that even though the minimum temperatures fluctuate in each year, that you can see that the freeze-tolerant eucalyptus continues to grow, even under the most severe freezes that this area has received. Even when controls, which is seen in the blue line here, are totally knocked back to the ground. Basically, there is a little bit of height loss on the freeze-tolerant eucalyptus, but it's able to grow back, as you can see on the right, in the spring. Eucalyptus is not a native of the U.S., and therefore there are no sexually compatible native species for it to breed with. The hybrid that we're using has limited natural reproduction. Because it's a hybrid, it has inbreeding depression, so that it doesn't cross easily with itself. It has poor seed germination, and it doesn't propagate. You know how some trees are able to drop their twigs and sprout? Well, eucalyptus is not able to do that. It can only be propagated through rooting by the human interventions in order to facilitate that process. So there's no chance of vegetative spread. We also have, as an extra measure of control to prevent any outcrossing from our transgenic eucalyptus, have introduced a gene, a pollen control gene. And in the right-hand side, you can see here, these are the anthers that are found in eucalyptus flowers. Here on the right, you can see a control, the transgenic, in which we've introduced a gene that stops pollen grains from developing normally. And the anthers here are totally pollenless, whereas you can see in the control, the anthers are just plump with pollen, and you can see little pollen grains on the surface. We've demonstrated this complete effectiveness of pollen control in multiple field trials and multiple eucalyptus trees. It's a very robust and stable trait, and we feel very confident that gene flow from these trees is prevented. We know that because eucalyptus is not a U.S. native, that there are environmental considerations. One of the considerations is land use. Eucalyptus plantations, we believe, will be used in the southeastern U.S. in a responsible way, and will be planted on land already operationally in pine plantations, or on marginal land in which agricultural crops are not currently being grown. Many people have concerns about eucalyptus being an invasive species, but when you say the word eucalyptus, what we don't really realize are there are about 500 species of eucalyptus. And so that the species that we're using, the Grandis biurophylla hybrid, that particular genotype, actually has very little in the way of invasive characteristics. And in fact, the University of Florida IFAS group that looks at invasiveness has scored the genotype that we're using for this work as a score of 3 out of a score of 10, which gives it the designation as non-invasive. Another concern that people might have about eucalyptus is that how much water does it use? And basically, eucalyptus uses less water than an agricultural crop does, such as cotton or soybean or corn. It probably uses, because of its fast growth rate, and water use is proportional to growth rate, might use more water than, as any hardwood might, than a slower growing tree. But it actually uses less water per unit weight of biomass. It actually, from a water use efficiency point of view, per ton of wood produced, eucalyptus uses less. So a short rotation eucalyptus harvest under coppices is not expected to have detrimental impacts to the water table or the aquifers. Another advantage of purpose-grown eucalyptus is that it can be grown cost-effectively. As I mentioned before, hardwoods are not grown in plantations today, and people might say, why is that? We grow pine in plantations. Why isn't hardwood grown in plantations? Well, that is because it's very difficult to establish hardwood plantations. And the cost of establishment is often too great for the productivity of the wood that's produced from some of the slow-growing trees, such as oak. So it's not really sustainable. So if you grow eucalyptus in a plantation, and you can see the arrow here, this is a study done by North Carolina State, the Wood to Ethanol Consortium. You can see the total cost of growing eucalyptus is basically $63 per bone-tried ton, but basically it's similar to now harvesting of natural hardwood, and it compares favorably with loblolly pine. So in other words, instead of being expensive like hybrid poplar or poplar cottonwood might be because of the cost of growing and establishing these and their slower growth rate, eucalyptus actually can be grown and bought at a level that allows the economics to work for both the landowner and the processor. So we talked about productivity and the improvements that could be made in productivity, but I wanted to mention another way to think about productivity is how do you get more out of the wood that you harvest? In other words, so do you have to harvest all that wood in order to get the end product? And in the case of making paper, sometimes the amount of lignin, which is a polymer within the wood that binds the cellulose fibers together in the plant cell wall, sometimes that lignin prevents the cellulose, which is used and turned into the pulp, from being processed efficiently. And so in order to get rid of the lignin, sometimes you have to almost pulp away half of it or a third of the actual cellulose. So if you can actually modify that lignin, you actually can perhaps do a processing in which less wood is needed to generate the same amount of pulp. And so in a way, this is another element of productivity, which is the downstream productivity. And so what we have looked at is you see a complex pathway in front of you here, and you have the end products down here of lignin, the guaiacyl lignin and the thuringial lignin, which both of these types of lignin are found in the cell walls of wood. And there are major enzymes involved in this highly complex biosynthetic pathway. But at the very start of the pathway, there are two different enzymes that have major rate-limiting capabilities, and that's an enzyme called C4H and an enzyme called C3H. And what we've done is we've looked at can you reduce lignin through reducing the enzymatic activity of C3H and C4H. And then what is the impact of this reduction on the processing capabilities? So we've shown in this graph here, you can see here marked in green are eucalyptus trees in which we've downregulated that rate-limiting step of C4H. And we can see that we can decrease lignin content by as much as 20%, but at the same time maintaining normal growth and vegetative features. These other orange were downregulated by the other approach, the C3H downregulation approach. And you can see that even though you see similar or even less lignin content, it has a much higher impact on growth. So we believe that the C4H gene gives that perfect medium between economically important reduction in lignin but maintaining normal vegetative growth. If you then look at the eucalyptus that has had C3H and C4H downregulation, you can see that they have similar and slightly higher carbohydrate contents. Here's the sugar percentage, and you can see the C3H mediated reduced lignin trees in eucalyptus, again in the C4H. And they all have fairly similar, but slightly higher perhaps than the control. But what we see is that the total sugar content or cellulose content in this wood is about 50 to 60%. However, if you're trying to remove lignin, if you're trying to get an increased amount of cellulose or an increased amount of cellulose that is then able to be converted into sugars, which then can be converted into biofuels, you can see that if you're a C4H mediated lignin reduction, you are able to release your sugars easier than if you're a non-lignin reduced tree, or if you've been reduced by the other pathway through the C3H lignin reduction approach. So what we've seen is that in the control, we've had, with a slight pre-treatment to remove the lignin, we've seen that we can get a 40% release of sugar, but in the case of the C4H, we are able to get up to 99% sugar release from the cellulose that's been made available by having the lignin reduction, which compares favorably, quite favorably to the control. And it actually compares quite favorably to other feedstocks that could be considered for biofuels applications, such as sweetgrass, in which you see sugar releases of 42 to 55%, and in the case of alfalfa, 45 to 80%. And these are transgenic lignin modified switchgrass in the case of where I'm pointing here, the switchgrass COMT transgenic, which is another lignin downregulated method, and in the alfalfa, the various is all transgenics where they've downregulated lignin. So we can see here that wood has the potential, in particular eucalyptus wood, to be a highly preferred feedstock for biofuels production. There is a strong correlation between lignin content and the total sugar release. You can see there's a linear relationship. Basically, sugar release is higher as the lignin is lower. So based on all this, we think a C4H lignin-reduced eucalyptus is a great feedstock for candidate for biofuels. It probably would be a great feedstock also for the pulp and paper industry. The low lignin mediated by C4H reduction greatly reduces the recalcitrance of biomass in being converted into biofuels. Sugar release correlates well with glucose content and lignin content. And since the C4H reduced eucalyptus grew similar to the control, its output could be 10 dry tons per acre per year in wood production, but also the output of 1,000 gallons of fuel per acre. So that is some kind of the upside potential of this tree. We can look at lignin reduction in another way, and this is an application that's focused at the pulp and paper industry. And this is where, as I mentioned before, the cell wall has within it two different types of lignin, the guaiacyl lignin and the syringal lignin. And what they've discovered in the pulp and paper process is that syringal lignin is bound less tightly to the cellulose, and it's easier to remove than is the guaiacyl lignin. So the idea is how can you increase the proportion of the syringal lignin to the guaiacyl lignin in the cell wall. Now there's a way of doing, there's an enzyme called the Cal5H, or some people call it the F5H enzyme, that's involved in converting basically the precursors that are directed towards guaiacyl lignin production and shifting those into syringal lignin production. So we tested to determine if you overproduce this enzyme so that the pathway shifted its movement and flow into the syringal lignin pathway, you might increase the amount of syringal lignin relative to guaiacyl lignin. And why that is important is because the kraft pulping process that is used to make paper requires a lot of chemicals, sodium hydroxide and sodium sulfide. So the idea is, can we reduce the amount of chemicals that it requires to remove the lignin? And we believed and tested the hypothesis that this low lignin or modified lignin with a higher SG ratio, a higher proportion of lignin that's more able to be removed or more reactive, what impact would that have on pulp yields as well as energy and chemical requirements to make paper? So we produced the transgenic eucalyptus plants that had an overexpression of the cal-5H gene, the coniferol 5-hydroxylase. And what we found was, in this graph here, you can see there's three different types of trees being tested. There's the control in red. There's two different ways of expressing or overexpressing the cal-5H gene. So one is stronger than the other. You can see this one in blue right here gave us the strongest effect. And you can see the green one still improved SG ratio, but didn't give the same strong effect as did this cal-5H2 version of the gene. So we've been able to show an improvement in SG ratio from an average of 2.3 in the range of basically 3 to 4.6. What we saw that if you increase the SG ratio, the syringal lignin to gliaceal lignin, basically what you would do is you increase the amount of soluble lignin relative to insoluble lignin, which means that you're increasing the amount of lignin that's more easily removed. And in the process, if you increase the SG ratio, you can see less chemicals are needed. Here's active alkali, or the sodium bisulfate that we talked about before. You require less of that active alkali chemical in order to produce pulp. So at the higher SG ratios, less chemicals are needed. And we actually saw pretty much an average of a 15% reduction in chemical cost. And chemical use, obviously, because cost is related to the amount of chemicals used. And then the great thing about it, you're using less chemicals to do the process, but you're still being able to increase pulp yield. So as you have less use of chemicals, you have a higher pulp yield. So you've had multiple benefits from here. You need less wood to get the same amount of pulp. You're making it with less chemicals. So it's a great win-win for everyone in terms of sustainability as well as for the processor. So in summary, basically looking at this eucalyptus example, we see that biotechnology can provide benefits for renewable bioproducts from wood, whether they be for the pulp and paper industry or for the bioenergy and biofuels industry. We believe that fast-growing short-rotation eucalyptus provides a biomass crop which can generate up to 33 green tons or 16 dry tons per acre per year. We believe that the addition or testing of cold tolerance in this tree enables the possibility of growing this highly productive and valuable crop in the southeastern U.S., thus by helping maintain the forest industry and an important economic driver in the southeastern U.S. Reduced lignin enables cost-effective economics for biofuel generation from wood, enabling the biofuels industry in the southeastern U.S., and altered lignin increases yields and reduces the cost of generating pulp. Another sustainable feature that's really important and again helps sustain the economics of the southeastern U.S. that is dependent on many pulp mills for a rural development and support. So that's what I have today. As I mean, our slogan, ArborGen's slogan is more wood on less land. So in other words, less land required to produce the wood that we need as a renewable feedstock. And that through our products, we'll see a more sustainable bioproducts industry that has a less reliability on petroleum-based products. So thanks very much for your attention. I can entertain some questions.
Thank you, Maude. We do have a few questions. I'll put those on the screen now, and we can try to address those.
Okay, number one, what it was the first biotech plant from 1996 you mentioned. That basically was the Flavor Saver tomato, and then closely followed by the Roundup Ready soybean. The second question is, what efforts are being made for greater acceptance of biotech crops? There are multiple organizations that provide information on biotech crops, and one of them that I mentioned before, ISA, I think is one of the most influential. But I think it's ISA is the International Institute of International Acquisition of, I forget it, I'm sorry, but if you type in ISAAA on and Google it, you'll find their website. And it's basically information dispersal, I think, is the most important thing, that people are transparent and open and communicating. And if people have questions, to direct those questions to people who have answers. And people in the biotech area, whether they be academics or in companies, are usually very happy to explain the technology and talk about it and indicate, you know, that they are working hard to bring about technology that addresses the needs of the industry, but also addresses the concern of society. Third question, what is the difference between direct and co-firing for producing electricity? Well, direct firing is when you don't use coal, you just use wood, and you're usually generating electricity through the generation of steam or heat. And in the case of co-firing, you're actually using wood alongside coal in a coal-burning plant. So it's a way of coal-burning plants of reducing their carbon footprint and having a cleaner burn by using wood. Fourth question, has the pulp and paper industry been able to see a difference in GM trees as opposed to naturally grown trees as far as quality? It seems that the yield is greater, but is the quality the same or better? As you know, you may not know that, but there are only a few commercially available biotech trees. One of them is a papaya that's basically in Florida and in Hawaii, and also in China they have insect-tolerant populus. There's another tree in the U.S. that is going through regulatory approval, and that is a virus-resistant plum tree. In our case, our trees have been tested extensively here, and that's an important thing that we do is when we do the field trials, we're introducing a trait, say, for example, for freeze tolerance. But we know that the tree is going to be used for a process, whether that be for pulp or paper or for energy. So it's very important that the tree perform exactly or better than what it did before. So in our tests, we don't just say, is it cold tolerant? We also ask the question, does it have the same disease resistance? Does it produce wood that's just as useful or better as the wood that a tree without the gene would produce? So those are all questions we would test in doing our field trials. And so it's important that in making a selection to go forward for actual use in the field for actual plantations, that it meet the industry's needs, the growers' needs, and that it hasn't obtained any other features that we would consider deleterious, like being more highly productive more than it was before, or something like that. So they go through a very intense selection process to identify a tree that is exactly or better than the tree that it was before, with the exception of that one introduced gene. It is a question in general, do faster growing trees, in the case of pine, have poorer wood? And there is a dogma that is true, but it's not necessarily linked one-on-one. And so there's genomics breeding programs as well as biotech programs that are trying to de-link that situation we see in pine, in which the faster growing trees, you start harvesting them sooner, the wood might not be as good in quality for structural lumber as it was before. But that, we believe, is something that will be attacked through a traditional breeding as well as through biotech approaches. On the freeze tolerance map you displayed, the westernmost border also is where there is a drier climate. Is this a factor in addition to cold? That's an excellent question from a scientific perspective, because the stress tolerance pathways between cold tolerance and drought tolerance or water, basically water deficit tolerance, they are closely related. Our gene that we introduced for freeze tolerance does confer a very slight improvements in water deficit tolerance, but not significantly so. So you're right, that it's a concern. Eucalyptus basically will be able to grow only when there's water available, and if there's no water available, it basically has the ability just to kind of wait. So it's one of those species that grows in the presence of water and basically slows down when there isn't water. 6, what is the educational level that our employees require? Well, we have, as I mentioned before, it's a company that's very diverse in terms of what we do. We have people who work in the field, we have people who work in the lab, we have people who work in the finance area, people who work in business development. We have people in nurseries and orchards that are propagating trees and putting them into containers and growing thousands and thousands of trees and crops. And so we have a wide diversity of the type of people that we bring in. There are people that work in the lab that have basically associate's degrees all the way through to PhDs. Some of our contract technicians, where we work through a contract employment agency, oftentimes just have a high school degree, but we train them here on site. So we have a broad level of different backgrounds that we utilize within the company. Question 7, how can the BioNetwork assist in training of new or existing employees? Well, we're always looking for great people, and I think that if the BioNetwork can stimulate people to get hands-on training in the laboratory areas, such as doing biochemistry or molecular biology or doing plant tissue culture, or just any hands-on laboratory experience is really welcome. Having a great education in terms of book learning is fantastic because it really helps in the problem-solving area, but we find that people who have had an opportunity to do hands-on work in a research lab environment are really the very best candidates. Question 8, have you considered how to engineer the eucalyptus that is less likely to burn? I think eucalyptus and pine have similar characteristics in their burnability, basically, and their combustibility. I think that some of the eucalyptus that people have reported have been an issue with fires are those in which the leaf litter doesn't decay rapidly. I think the approach that would be taken in those cases is to try to reduce the oil content in the leaves, the aromatic oils that are found in eucalyptus. Question 9, once, if a GM eucalyptus receives regulatory approval, how long will it take to scale up seedling production to meet commercial needs? Actually, what we do is anticipate, just like we do in our normal normal seedling sales. We start building, for example, the pine crop for 2012. We're actually starting to make, the pine crop for 2012 we started in 2010. So in terms of when a biotech eucalyptus would reach the market, we would have anticipated the regulatory approval based on the feedback we were getting from the regulatory agencies, and would have started doing the production of that particular crop at least 2 years before, or a year and a half before. Number 10, would these trees be grown on federal, state land or on privately owned property for the pulp, paper, biomass industries? And are these growers generally accepting of GMOs? Well, we believe that these trees are primarily going to be going to be grown on privately owned property by people who first will be adopted by people who are interested in being able to see that their land is returning to them something on their investment in a shorter period of time. So you'll see these trees first being adopted by people who want to be able to fund their children's education, for example, instead of their grandchildren's education, or people who are interested in basically increasing the profit that they have from their land. These biotech trees probably will first be grown for dedicated processors. So you can imagine that the earliest adopters would be some of those pulp mills or bioenergy facilities in which they need hardwood to supply, and they can see that the chances of them being able to have a smaller land footprint requirement for their process, that they would then be engaged with the local landowners to help them supply their mills. So we're anticipating that the landowners, in conjunction with the processors, will jointly adopt the technology in order to achieve both of their end goals simultaneously. Question 11, is ArborGen willing to work with us, BioNetwork Capstone Center, to develop a plant tissue culture course? We have expertise within our ranks and would love to be involved in this endeavor. Of course, we would love to help. We have students coming here all the time. We host all sorts of workshops. We have local teachers coming through all the time. The staff here at ArborGen just love to interface with teachers and with students. So we would be happy to help out in whatever capacity you would like us to.
All right, I believe that's all the questions. As we wrap up today, please take a moment to answer the brief survey on the right side of the screen. Your feedback will help us as we plan future BioForum events, and then just be sure to click Submit when you're finished. While you're doing that, note that today's presentation was recorded, so you can view it again. You'll receive a thank you email from BioNetwork that will include a web link to the recording. And you can find out about other BioForum events by visiting our website, ncbionetwork.org. And I ask you to go ahead and mark your calendar for our next BioForum. Registration for this event is free and available online at ncbionetwork.org. Well, just before we close up today, Maude, do you have any final comments or anything you'd like to share with us before we wrap up?
No, it's been a real pleasure, and if anybody has any further questions, I'd be happy to entertain them. You can be free to pass on my email address.
All right, we'll do that in the chat box just momentarily. Thank you, Maude, and thanks also to BioNetwork for making this presentation possible. And thanks, of course, to you for your time and participation in this webinar. On behalf of BioNetwork, thank you and have a great day.